All right. Sorry, guys, was managing logistics for the other rooms. But welcome to room 5 of the SRI Symposium. Glad you guys are in my room. I hope it's going to be the most fun, awesome room. We are going to get started with our first presentation from Wenqing, who's going to be talking to us a little bit about virus host evasion. Take it away.
Okay, hello, and thank you for taking the time to review my project. The topic I chose to focus on is the species-specific immune evasion strategies employed by the dengue virus. Dengue virus is an RNA virus transmitted through mosquito bites, and as someone who grew up in tropical regions, I have seen the challenges posed by mosquito-borne diseases since childhood. Among them, dengue stands out as a major public health concern and can result in lethal and severe symptoms. It infects approximately 400 million people annually, with more than half of the world's population living in areas at risk. And the geographic range continues to expand due to climate change and population mobility. Despite its global impact, treatment options remain limited, and current vaccines only offer partial protection. One of the major obstacles in developing effective vaccines and therapeutics is the lack of accurate animal models. Non-human primates or NHPs are commonly used due to their genetic similarities to humans, but they exhibit mild or absent with often with lower viremia or the concentration of viral particles in the blood. Why? My review suggests that there's key differences in innate immune responses that underlie these discrepancies, particularly noting two critical pathways, cGAS-STING and JAK-STAT, and how dengue has evolved to antagonize key components of these pathways in humans but not non-human primates. The cGAS-STING pathway is typically activated by cytosolic DNA and serves as a critical driver of interferon or IFN production and interferon-stimulated genes or ISG expression, helping establish an antiviral state. Although dengue is an RNA virus, it activates this pathway indirectly by mitochondrial DNA leakage into the cytoplasm caused by viral encoded proteins. In humans, dengue cleaves STING using its protease. However, this cleavage does not occur in non-human primates or mice due to subtle amino acid differences at the 78th and 79th position of STING. Human STING contains an RG motif at the site, making it susceptible, whereas NHP and mice encode alternative residues that allow resistance. As a result, interferon responses are suppressed in human cells but preserved in non-human primates, limiting the virus's ability to replicate in animal models. The JAK-STAT pathway is another key antiviral mechanism. It transmits interferon signals from infected cells to neighboring cells, preparing them to resist infection. In this pathway, secreted interferons triggers phosphorylation of STAT1 and STAT2, which then forms complexes called ISGF3 that drive ISG transcription. In human cells, dengue effectively inhibits STAT1 phosphorylation. However, this inhibition fails in NHP cell lines. Moreover, STAT2 alone can drive antiviral response even in the absence of STAT1, but dengue also targets STAT2 in humans. The impact of dengue on STAT2 in non-human primates remains elusive as no direct studies have been conducted. But what we do know is that mice engineered to lack both STAT1 and STAT2 exhibit lethal disease. This suggests that STAT suppression is critical to severe disease and may explain why humans experience more severe outcomes than non-human primate models. So could dengue benefit from lower viremia in non-human primates? I believe so. Dengue circulates in the endemic cycle involving humans and mosquitoes, and the sylvatic cycle involving non-human primates and mosquitoes. Dengue is believed to have originated from sylvatic origins, yet both endemic and sylvatic strains are capable of cleaving human sting while leaving non-human primate sting intact. And dengue is more effective in humans. One explanation may lie in host density and transmission dynamics. In sylvatic settings, where non-human primate hosts are sparse, dengue might benefit from a longer, lower-level infection to increase the chance of mosquito acquisition. In fact, studies show that asymptomatic cases, often characterized by lower but prolonged viremia, can still effectively transmit to mosquitoes. In contrast, humans live in dense populations, reducing selective pressure to prolong infection. In this context, dengue may have evolved to maximize replication and spread rather than preserve host survival. These findings have important implications for animal model development. Priming non-human primates with interferon inhibitors prior to infection or creating transgenic mice expressing humanized STING could be possible strategies, and ultimately, dengue's ability to evade human but not animal innate immunity helps explain why modeling the disease has been so difficult. A deeper understanding of species-specific immune response is essential to improving research and moving closer to effective vaccine and treatment strategies. I'd like to thank the SRI staff for making this opportunity possible, and especially my mentor, Courtney, for her guidance. She made this experience unforgettable, and it will undoubtedly shape my future aspirations. Thank you.
Awesome. Love to see it, and thanks for that. Now we'll have Kashfi talk to us a little bit about what she developed with Proteotag.
Okay, so hi, my name is Kashria Stogie, and I'll be talking about my computational research project, Proteotag, which is a rational approach to labeling self-assembling protein nanoparticles. Designed protein nanoparticles were first introduced in 2001 by Padilla and her colleagues, who fused unique oligomeric proteins with a linker for self-assembly and symmetry. Recently, tools like Rosetta and AI/ML software such as Protein MPNN have been used to redesign these particles, improving their interfaces. These advancements are vital in structural and cellular biology and vaccinology. In structural biology, nanoparticles provide platforms for studying protein structures and interactions. In cellular biology, they can be engineered for targeted drug delivery and used as imaging agents. In vaccinology, they enhance immune responses and serve as scaffolds for antigen display. Notable examples include T3351, which enabled high-resolution cryogenic electron microscopy, and I5350, an immunogen against pathogens like the influenza virus. Despite these successes, the diversity of nanoparticles is limited, and no machine learning models are trained specifically for their design. Existing methods rely on an iterative cycle of backbone generation and refolding, needing large datasets that preserve symmetric interfaces. This is something that current protein data bank structures lack. This is shown in Figure 1 in the bottom left corner, illustrating inconsistent chain labeling in homomers 1DAT, 9FO3, and an unpublished 6-gen, and heteromers 5CY5, 5IM5, and 7SGE with arbitrary IDs. So for our method, we developed a Python script using PDB and MMSIF parser for analysis, MMSIF IO for SIF output, and select from BioPython for manipulation, with NumPy support for numerical operations. The code classified chains as heteromeric or homomeric by amino acid sequences. Homomers use A for the first chain, while heteromers use uppercase A and lowercase a. Average atomic coordinates define the centroids and then classify neighboring chains within 25 NumPy units. Angles between centroids indicate trimeric, tetrameric, and pentameric interfaces, with dimers assessed separately. Figure 3 shows selecting an origin, finding neighbors, classifying symmetry, and relabeling. The final scheme in Figure 2 labels the central chain as A and surrounding dimers as B, trimers as C and D, tetramers as E and F, and pentamers as G and H, with lowercase for the second subunit and heteromers. Figure 4 shows all 6 nanoparticles from Figure 1, where all interfaces are correctly relabeled, even with overlapping symmetries. This system enhances clarity with non-arbitrary labels, aiding researchers in integrating AI/ML for designing novel nanoparticles by providing structural information. However, it heavily relies on interfacial symmetry, overlooking other biological factors like intermolecular forces, such as hydrogen bonding and salt bridges. Low resolution structures may hinder interface detection, and untested nanoparticle designs might also need further adjustments. Despite these concerns, Proteotag is taking a promising step towards the rapid design of novel nanoparticles. In future work, we will expand our test set to include a wider variety of self-assembling protein nanoparticles and complex macromolecular structures. This will enhance our understanding of proteotag and enable physics-based calculations with Rosetta to investigate energy differences in protein-to-protein interactions. We also plan to develop AI/ML models to classify interfaces of predicted structures, using AlphaFold for negative controls and existing protein data bank structures as positive controls. Lastly, I would like to thank Harvard's undergraduate Open Bio SRI for the opportunity to conduct this research and Morgan G for his invaluable mentorship. Thank you for listening.
Awesome. Great presentation. Uh, Now we will move on to Shreya, who will tell us a little bit about glioblastoma treatment.
Hello, everyone, and thank you for being here. My name is Shreya Shetty. Today, I'll be presenting a systematic review that discusses ferroptosis-inducing mechanisms in glioblastoma treatment. Glioblastoma, or GBM, is a highly aggressive brain tumor with a very poor prognosis. Even with standard treatments like surgery, radiation, and chemotherapy, most patients experience recurrence, and the median survival time remains just over a year, largely due to the tumor's intrinsic resistant mechanisms. Ferroptosis is a recently identified form of regulated cell death that's iron independent and characterized by the accumulation of lipid peroxides and oxidative membrane damage. It differs from apoptosis and necrosis because it has distinct molecular drivers like GPX, lipid metabolism, and iron homeostasis. Interestingly, GBM cells often have dysregulated iron metabolism, making them potentially vulnerable to ferroptosis. Recent research has focused on strategies to selectively induce ferroptosis in GBM cells using both pharmacological and genetic tools. This review aims to systematically evaluate the current literature on ferroptosis in GBM, including drug targets, RNA regulators, and iron metabolism pathways. To conduct this review, we performed a systematic literature search using the PubMed database. We used mesh terms like ferroptosis and glioblastoma and lipid peroxidation and glioblastoma. After rigorous rounds of screening, a total of 39 articles of the original 151 research articles were included in the paper for detailed analysis. These articles were also sorted into three main groups that could be used as therapeutic targets. One major strategy to induce ferroptosis in GBM cells is through natural and synthetic compounds. Natural agents such as CBD, genisenide RG5, and procyanidin B1 have been shown to trigger ferroptosis by decreasing antioxidant capacity and altering iron metabolism. On the synthetic side, compounds like phytostatin, BH3 mimetics, haloperidol, and S670 target multiple pro-survival pathways. These drugs typically work by inhibiting GPX4, activating the AKT/mTOR pathway, or suppressing NRF2, which is a regulator of antioxidant defenses. Several of these compounds also improve the efficacy of TMZ, which is the standard chemotherapy for GBM, by reducing the tumor cell viability and stemness. Another emerging area of interest involves non-coding RNAs, particularly circular RNA and long non-coding RNA. These RNAs regulate ferroptosis-related genes at transcriptional and post-transcriptional levels. For example, circle RFN5 acts as a tumor suppressor by promoting ferroptosis and reducing GBM stemness. And in contrast, circle RFN10 and circ VPS8 inhibit ferroptosis and are associated with tumor growth. Among long non-coding RNAs, link 02587, DLEU1, and link 01088 have been shown to suppress ferroptosis through various mechanisms such as sponging miRNAs or altering transcription factor activity. A third important mechanism involves iron and lipid metabolism. Since ferroptosis is driven by iron-independent lipid peroxidation, many therapeutic strategies aim to exploit this vulnerability. COPZ1 is one target of interest because it's overexpressed in GBM and associated with poor prognosis. When COPZ1 is knocked down, it increases transferrin receptor and NCOA4 expression, enhancing iron uptake and ferritinophagy, which together promote ferroptosis. However, COPZ1 also plays a broader role in vesicle trafficking, and its inhibition could disrupt other cellular processes, raising concerns about toxicity. In summary, ferroptosis represents a promising therapeutic avenue for glioblastoma by targeting its vulnerabilities in iron metabolism, antioxidant defenses, and RNA regulation. While still in early stages, these findings offer a foundation for developing more effective and targeted treatments for glioblastoma. Lastly, I'd like to thank the Harvard Open Bio staff for providing us with this amazing opportunity. And I would also like to thank my mentor, Jae-yoon Shu, for her guidance and mentorship throughout this process. Thank you.
Awesome. Great presentation. We'll now go to Aiden, who will tell us a little bit about myeloid leukemia.
Hey everyone, I'm Aiden, and today I'm excited to share my research on how cannabidiol or CBD could play a role in treating acute myeloid leukemia through lipidomics and network pharmacology. So before we begin, let's talk about acute myeloid leukemia or AML, which is an aggressive blood cancer caused by genetic alterations in hematopoietic stem cells. It accounts for about 1/3 of all adult leukemia cases, has high relapse rates, and many older patients can't tolerate intensive chemotherapy treatments for it. So there's a real need for new targeted therapies. Recent research shows AML cells have dysregulated lipid metabolism, meaning they produce and use fats abnormally. Lipids aren't just passive fats, they're active players in cell signaling, membrane structure, and apoptosis. Apoptosis is an important term, and it's basically the programmed death of damaged or cancer cells. When lipid metabolism goes wrong, cancer cells can evade treatment and fuel their own growth. CBD, the non-psychoactive compound from cannabis, has been shown to affect lipid signaling, alter membrane composition, and trigger apoptosis in cancer cells. However, the molecular details of how this happens in AML are poorly understood, and that's where our study comes in. We use lipidomics, a branch of metabolomics that profiles all the lipids in a system, together with network pharmacology, which maps how a drug interacts with genes, proteins, and pathways. This combination lets us see which lipids are changing after CBD treatment and connect those changes to the genetic networks that might be causing them. And the goal of our research is quite simple. It's to figure out the mechanisms by which CBD alters lipid metabolism in AML cells and identify both lipid biomarkers and molecular targets that could explain its therapeutic potential. Now let's transition to how we did our study. First, HL60 leukemia cells were treated with CBD at 10 micromolars for 48 hours by the original investigators. We then accessed this lipidomics datasets from Metabolomics Workbench, pre-processed the data, and ran statistical analyses in MetaboAnalyst. For network pharmacology, we collected CBD targets and AML-associated genes from public databases, identified overlaps, built protein-protein interaction networks, and performed enrichment analyses. Now for the results. Let's move to the top left of the poster for Figure 1. Figures 1A and 1B show two score plots, a PCA and OPLC score plot. Each dot is a sample, and the separation between red and green dots show that CBD drastically changes the lipid profile of these leukemia cells. Figure 1C is our permutation test for the OPLS-A model with Q squared equal to 0.981 and R squared Y equal to 0.995, which shows our model has excellent predictive accuracy. Lastly, Figure 1D is our volcano plot, which maps fold change against statistical significance or p-values. Here, the red dots are lipids that are more abundant in CBD-treated cells compared to the sample control group, which means they're upregulated, and blue dots are less abundant, which means they're downregulated. This gives us a short list of significant lipid biomarkers. However, it's not the full list yet. Moving to Figure 2, we dig deeper into these biomarkers for the full list. Figure 2A is a heat map showing the clustering of 81 significantly altered lipids, which was determined by univariate analyses. You can see clear patterns of up and down regulation between treatment groups. Figure 2B is our random forest analysis ranking of the top 15 lipid species most predictive of CBD treatment status. Figure 2C is the pathway enrichment bubble plot. Here, the two most impacted lipid pathways were sphingolipid and glycerophospholipid metabolism, with linoleic acid metabolism close behind. This is important to remember. Now that our statistical analysis is complete, we identified phosphatidylcholine, or PC, and triacylglycerol as our key lipid biomarker species. And remember these lipid biomarker species as well, as they will play into our cohesive story at the end. Now over to the right-hand side of the poster, Figure 3 is our network pharmacology analysis. The Venn diagram in 3A shows 88 overlapping targets between CBD and AML. The network plots in 3b and 3c reveal the connections between these overlapping targets, highlighting how genes, which are highly connected nodes. Enrichment analysis in 3b and 3e show the top gene pathways affected, which many of them were tied to membrane-associated signaling. Finally, let's bring it all together into that one cohesive story I was talking about, shown here in figure 4 at the bottom right. This is our proposed mechanism. One of our top hub genes was CYP3A4, a metabolic enzyme known to be inhibited by CBD. If CYP3A4 activity is inhibited, linoleic acid availability increases, which, if you remember, was a significantly impacted pathway. This excess linoleic acid feeds into phosphatidylcholine remodeling, leading to phosphatidylcholine accumulation in membranes, which, if you remember once again, phosphatidylcholine was a top lipid species biomarker. This shift in lipid balance could stress a cancer cell's membrane systems, disrupt signaling, and trigger apoptosis. To wrap it up, we identified key lipid biomarkers altered by CBD, linked them to major metabolic pathways, and proposed a CYP3A4 linoleic acid PC mechanism that could underlie CBD's anti-leukemia effects. Now what's left? Well, our sample size was small and limited to one AML cell line, which is why larger datasets and experimental validation will be essential to confirm these findings. With the time I have left, I would like to thank my absolutely wonderful mentor, Tomiwa Oedo-kun, who spent his late nights helping me with this project, and of course, a big thanks to the Harvard SRI program for inspiring the next generation of scientists. Thank you so much.
Awesome and great research, Aiden. Really appreciate the presentation. We'll now move on to Juan, who's going to tell us a little bit about removing contaminants from drinking water.
Good morning. My name is Juan, and I'm great to be here. Water is the most fundamental resource for life, yet its quality is increasingly degraded. Industrial, agriculture, and municipal activities release persistent micropollutants into our waterways, and conventional treatment methods are largely ineffective against them. The solution may not lie in more than synthetic chemicals, but nature itself. Our literature review explored biopolymers, materials sourced from abundant renewable biomass like cellulose and agriculture's cytosine from shellfish, alginate from marine algae, and lignin from the paper industry. These sustainable membranes can effectively target and remove modern contaminants, representing a critical step toward global water security. Biodegradable membranes have been researched for years, but interest has grown due to three key advantages over petroleum-based membranes. First, sustainable and circular sourcing. They're made from renewable biomass, including reducing the reliance on fossil fuels and repurposing materials otherwise considered waste. Second, inherent anti-fouling properties. Synthetic membranes are often hydrophobic, causing oils and microbes to clog pores. Biopolymers, on the other hand, are naturally hydrophilic, forming a water layer that resists fouling and maintains stable water flow. Finally, a biodegradable end of life. Instead of persisting as microplastics, these membranes decompose into CO2, water, biomass, creating a closed-loop system. To understand real-world performances, we reviewed only peer-reviewed studies from 2020 to 2025. The results were impressive. Heavy metals and alginate cytosine aerogel achieved a lead absorption capacity of over 13,031.6 milligrams per gram. Organic pollutants, modified cellulose membranes removed dyes of over 750 milligrams per gram, and advanced bioreactors achieved 100% removal of pharmaceuticals, macro scale separation over 98% salt rejection and desalination, and 99% oil water separation efficiency. These results raise a question. How are they engineered to be so effective? Synthesis methods like phase inversion and electrospinning create dense, selective layers of high surface area nanofibers. Characterization tools such as SEM and FTIR ensure pore structure and chemical composition meet design goals. Performance for further boosted by embedding functional nanomaterials. For example, graphene oxide for strength and permeability, metal organic framework, MOFs, to selectively capture contaminants, silver nanoparticles for antimicrobial protection, and the list goes on. This strategic engineering transforms simple biopolymers into multifunctional tools. However, moving from laboratory success to large-scale adoption will require addressing key challenges. For example, reducing production costs and ensuring long-term durability in diverse environments and creating regeneration methods that allow membranes to be reused. Despite these hurdles, the evidence is clear. Biopolymer membranes are not a distant dream. They are already a validated high-performance technology capable of tackling our most persistent water pollutants. Their great strength lies within their dual impact, purifying water while replacing petroleum-based plastics with a sustainable, biodegradable alternative. This approach is more than a technical solution. It is a critical step forward to global water security, built on innovations that work with nature rather than against it. That concludes my presentation. I'd like to thank the Harvard Open Bio program for this opportunity and my mentor, Tohib Obidara, for his guidance and support. Thank you.
Awesome. Great presentation. Hopefully, you're able to find a way to get contaminants out of my drinking water because that seems unfortunate. We'll now move on to Nirmal, who will tell us a little bit about decision drivers in cancer.
Hello, everyone. Is my audio fine? Just want to make sure. All right, sounds good. Good morning, everyone. Thank you for listening in. I'm excited to present my research on decision making in cancer treatment tourism. So, according to Lunt, medical tourism has grown rapidly in recent years because patients have begun to travel internationally to other destinations for care that is more affordable, timely, and higher and more personalized when it comes to quality. And usually, the care that they seek isn't available in their home countries. So while the majority of research in cancer and medical tourism has been focused on elective or wellness procedures, cancer treatment stands on its own because of its life-threatening stakes and urgency, which differs greatly from other types of medical travel. For example, unlike someone seeking cosmetic surgery, cancer patients often face longer wait times and also high costs and limited therapy options from home, and that can force them to make rapid and emotionally charged decisions about whether to travel abroad for treatment and what country they would like to go to seek treatment. Unfortunately, despite cancer's global prevalence, the literature in medical tourism has not fully explored the unique drivers in the decision-making process for cancer patients, specifically in the context of medical tourism. So that's where our study comes in. Our goal in the study was to understand how push and pull forces shape patients' choices. So, according to Side and Moriana's framework, push factors are conditions that drive patients away from their home healthcare systems, such as treatment delays and high costs, and push factors attract patients towards another country, and this can include factors like better technology or affordability. So when it comes to the methodology, our study employed the PRISMA SCR guidelines for adapting a bibliometric and narrative review. And we integrated the framework that was proposed by Arksy and O'Malley and Taj et al. To provide a comprehensive and complete overview of the topic of medical tourism related to cancer. So we also wanted to conduct a co-occurrence keyword analysis, which revealed three main thematic clusters. But to get that analysis, we had to first create a keyword cluster and query, which included keywords related to medical tourism, like cancer treatment and etc. And from there, we inputted that query into Scopus database, which gave us 878 articles. From there, we excluded articles and filtered them from stuff like data range. So articles outside that were published before 2016 and after 2024 were excluded. Articles that weren't written and published in English were excluded, and duplicates were also removed. And that resulted in an analysis corpus of 754. And then we specifically looked at cluster 6, which included 46 items and keyword items, which included keywords like cancer, oncology, neoplasm, palliative care, etc. And from there, we created a keyword-based document and thesaurus file, which integrated and enhanced the keyword document, and that removed over 79 duplicates. And that gave us a final corpus of 67, which we use for a bibliometric analysis. Additionally, from those 67 final articles that we gained, we also did further filter them out to 10 final articles for the narrative review. And we did that by looking at the abstracts and by deciding which articles were relevant to our study, specifically on cancer treatment. And we also utilized the Side and Mariana's push and pull framework to decide which articles and which literature base can also best decide and determine which could best determine the decision-making process based on push and pull factors for patients when it comes to cancer medical tourism. So the final co-occurrence keyword analysis revealed three clusters which focused on cancer-related topics. So the first cluster, which is the green cluster, focused mainly on demographics. The red cluster, which is the second one, focused on like cancer treatment procedures when it comes to like healthcare access and healthcare infrastructure. So it includes terms like cost, healthcare delivery, etc. And then the blue cluster focuses more on procedures. So it includes keywords like radiotherapy, chemotherapy, etc. So three clusters reveal a lot about the decision-making process and push and pull factors. So for example, the green cluster, which looks at demographics, finds that women and middle-aged adults were more likely to engage in medical tourism for cancer-related surgeries. And the red cluster also finds that push and pull factors related to infrastructure can help determine the decision-making process. For example, if your home country had less infrastructure and had higher cost, it could drive you to seek care in another country that probably had more personalized care, better technology, and more affordable care, which means that it was more attractive when it comes to like more personalized cancer care. And then figure two shows the push and pull conceptual model that we utilized in our specific research study. So figure two shows that this is a nonlinear process. So there are both push factors and pull factors that play a role. For example, push factors like long treatment delays and also high domestic costs can push a patient to seek care in another country, while pull factors like imagery and also perception of the quality of care can help attract patients to that country. And this is kind of a nonlinear approach that helps determine the emotional urgency and also the emotional decision-making process that the patient takes when deciding where and what specific cancer treatment procedure they would like to take and where they would want to take that. So the decision-making process can include steps like peer recommendation and online research, where patients quickly ask peers for recommendations of where to get procedure, and also they go through online research like social media, etc. And as you can see, this isn't the most viable because and there's are potential risks. For example, there could be complications after post-operation, and there could also be a lack of continuity of care because once you go to your destination and you come back, there could be a lack of treatment plans once you come back to your home country. So there are plenty of potential risks that still need to be studied and further analyzed. So to conclude, cancer procedures are a distinct thematic cluster when it comes to medical tourism literature, and patients' decisions are driven by push and pull factors. For example, push factors include limited local options, whereas pull factors include better care abroad. And so we included a narrative analysis which supports the push and pull decision-making model, which enhances the decision-making process that patients take when seeking and deciding where they want to take care abroad. And we also concluded that our study fills in gaps by combining quantitative mapping from the bibliometric review and also by utilizing narrative synthesis to explore gaps in cancer medical tourism like cost, accessibility, etc. And this study can be applicable in numerous scenarios. For example, it could help guide policy on cross-border cancer care, and it could also help support patient decision tools with push and pull insights. But obviously there are some limitations, and so future research is required. So we would like to validate motivations with empirical patient data, and we would also like to study regional variations in cancer tourism trends in the future. So with the remaining time I have left, I would also like to thank my research mentor, Jamie Garlington. She helped me a lot in this research, and she helped guide me throughout this process. Thank you.
Awesome. Thanks for that, Nirmo. Now we will move on to Kevin, who's going to tell us a little bit about deriving biomass-derived plastics.
Hello, my name is Kevin from Brentwood High School, and my mentor is Dr. David at Sunny ESF. Our project looks at the practical way to fight microplastic pollution by switching from petroleum-based plastics to biomass-derived plastics made from lignin, cellulose, and hemicellulose. But why does this matter? Microplastic pollution is accelerating fast. The world produces roughly 350 million tons of plastic each year, and tiny particles have been found in oceans, freshwater, soils, and even inside us. Right now, there are roughly 7 grams worth of microplastics in our brain. That's roughly enough to make a plastic spoon. But at the same time, we generate huge streams of agricultural and forestry residue, such as stalk, husk, and pulp mill byproducts. Those wastes are rich in 3 natural polymers: cellulose, hemicellulose, and lignin. Our central idea is to turn those into bioplastics that can reduce microplastics at the source while fitting better into a circular economy, as we are currently discarding 8 times more biomass waste than conventional plastics. That's roughly 2,830 million tons. I want to provide a quick overview of our biomasses. Cellulose gives strength and good barrier potential, while hemicellulose brings film forwarding and oxygen blocking capabilities. Lignin provides UV protection, antioxidant, and antimicrobial activity. Life cycle studies show that bio-based plastics can cut greenhouse gases and, if designed right, enable composting or upcycling at end of life. Recent reviews have highlighted that PLA production alone is scaling rapidly and that it could replace a portion of PET plastic applications and packaging. But what do we focus on? The first is blending for functionality. By blending PLA with lignin and cellulose and hemicellulose, we tune stiffness, toughness, and degradability. Lignin nanoparticles can add UV shielding and antimicrobial functions, while cellulose nanofibers reinforce mechanical strength. Surface modification and for compatibility was another of our focuses. Many of these biopolymers don't naturally mix. Surface chemistries like esterification or grafting plus nanofillers improve dispersion and interfacial adhesion, which boosts barrier and processing performance. The third is biorefinery integration. We emphasized extracting all three from the same biomass feedstock. Modern fractionation methods, such as alkaline processes, can recover high-quality streams that feed films, coatings, and compostable composites, minimizing waste and supporting circularity. There are also huge environmental and economic impacts. The first is greenhouse gases. Bio-based plastics can have a lower footprint than fossil fuel plastics when renewable power production and waste is composted or recycled. The second is microplastics. Moving packaging and films to compostable bio-based systems reduces the persistence of fragmentation. The graph below shows degradations of different types of plastics over the first 60 days. Please note that PET plastics did not degrade within the first 60 days, roughly less than 100%, while the bio-based plastics already began degrading, with some degraded by 50% by day 40. There are also severe policy and infrastructure implications. Labeling such as biodegradable or compostables can be confusing as there is no true global standard. Many regions still lack industrial and composting and dedicated recycling for bioplastics, so that standards and consumer guidance are essential to avoid greenwashing and contamination of waste streams. So what's the takeaway? Biomass-derived polymers are abundant, functional, and they're engineered and labeled, and if labeled responsibly, can replace a meaningful fraction of petroplastics, cut greenhouse gas emissions, and reduce microplastic generation. The science and materials are there, and the next step is scaling biorefineries, creating standards, and building composting and upcycling pathways so that these materials actually close the loop. Thank you so much.
All right. Thanks for the presentation, Kevin. We will now pass it over to Satvika, who will tell us a little bit about motor circuits in mammals and songbirds.
Hi, my name is Satvika Minavalli. Today, I'll be sharing my research focused on comparative neuroanatomy and neuronal diversification in vocal motor circuits, specifically in songbirds and mammals. Although much remains unknown about how neural circuits for fine motor skills like speech develop, songbirds offer a uniquely powerful model for studying these processes. Why songbirds? Because they are one of the very few groups capable of learned vocalizations, and their brain regions, especially the HVC and RA, which help control timing and send signals to muscles used for singing, closely resemble structures in the mammalian neocortex, which is the outer part of the mammalian brain that handles complex thinking, movement, and sensory processing. We conducted a literature review using peer-reviewed studies from PubMed and Google Scholar. We prioritized articles with clear methods and relevance to songbird motor circuits and their mammalian analogs. Data extracted included species, brain regions, neuron types, gene expression patterns, and experimental techniques. The findings were grouped into molecular, physiological, and evolutionary themes. One major discovery is that neurons in the HVC and RA share molecular and functional traits, with those in the mammalian neocortex and ventral pallium, which is a part of the embryonic brain that later forms motor and cognitive regions. This supports the idea of convergent evolution, that birds and mammals independently developed similar neural solutions for complex vocal behaviors. Figure 1 shows a visual comparison of the songbird and human brain, highlighting similarities in vocal motor pathways. In Figure 2, we see three major hypotheses explaining how song-related neurons develop. Figure 2a suggests immature neurons are molecularly programmed for either song or non-song functions. Figure 2b proposes gene duplication as the origin of song circuits. Figure 2c shows that song and non-song neurons may have matching molecular profiles and parallel circuit outputs, pointing to an evolutionary recycling of motor pathways. Another critical finding is the role of sensory feedback. In both birds and humans, vocal learning relies on feedback to maintain stability. When birds are deafened, their songs destabilize, accompanied by changes in brain circuits and gene expression. Social interaction and tutoring are also vital. And environmental deprivation can result in abnormal vocalizations. Understanding how vocal motor circuits evolved and function in songbirds doesn't just teach us about birds. It also sheds light on human speech, language development, and even disorders like apraxia or stuttering. By studying shared genes and pathways, this research may inform therapies or interventions for speech and communication challenges in humans. Though molecular tools are rapidly advancing, challenges remain, such as small sample sizes, species variation, and fewer genetic tools for birds compared to mammals. That said, new techniques like single-cell transcriptomics are helping us explore previously unidentified neuron types involved in vocal learning. Ultimately, this research helps us understand not just how birds learn to sing, but how complex motor behaviors, like human speech, may have evolved. By studying both conserved and convergently evolved traits, we move closer to answering big questions about brain development and behavior. Thank you for listening.
Awesome and great presentation, Satvika. We're now going to move on to Leon, who's going to tell us a little bit about microRNA regulation. Take it away.
Hi there. My name is Leon, and I'm really excited to share my research with you today. So, I've had eczema for as long as I can remember. Now, if you've experienced it or know someone who has, you know it's more than just dry skin. It's unpredictable flare-ups, constant itching, and a constant search for treatments that actually last. Living with eczema made me curious about why it happens at the cellular level and whether we can target those processes more precisely. That personal connection is what inspired me to investigate the biology of atopic dermatitis, or AD, and explore how RNA-based therapeutics, specifically microRNAs, might be able to help. So just a little bit of background. AD is a chronic relapsing skin condition affecting about 20% of children and 10% of adults in developed areas. Treatments like creams and biologics can work well, but many patients relapse within just a few years. What's still missing in research is a deep understanding of the skin's vascular and immune microenvironments and how that drives disease. One interesting gap in knowledge is how the blood vessel cells in the skin, known as endothelial cells, might influence the epidermal cells that form the skin barrier. We suspected there might be an intermediate cell state connecting the two, something not widely documented in eczema before. And to explore this, we analyzed single-cell RNA sequencing data from more than 40,000 skin cells, covering both healthy skin and AD lesions. Using a method called trajectory analysis, we mapped out the development paths cells take. This revealed a previously unrecognized vascular to epidermal cell state transition, connecting endothelial to keratinocytes. Next, we identified the key genes that change along this transition and searched curated microRNA mRNA databases for known microRNAs regulating these genes. This pointed to well-known regulators like the LET7 family and MER34A5P. But we didn't want to stop there. We wanted to go beyond known biology and design new synthetic microRNAs that could precisely target this transition. For that, we trained a deep neural network by the name of a variational autoencoder on over 7,600 human pre-microRNAs. We then generated 20,000 synthetic candidates, filtered them for stable hairpin structures, and prioritized those with strong predicted binding to our transition-specific genes. So on the poster here, right in the middle, you can see a UMAP plot showing clear separation of major skin cell types, vascular endothelial cells on one end, and keratinocytes on the other, and this transitional line in between. The heat maps below that show the top 25 genes upregulated in vascular endothelial cells and in keratinocytes during the transition, highlighting pathways like adhesion, barrier repair, and immune regulation. For the microRNA analysis, the known regulators we identified have strong links to vascular remodeling and keratinocyte differentiation, validating that our computational pipeline is on the right track. And from the generative model, we narrowed our synthetic pool to highly stable seed unique candidates, meaning they're less likely to have off-target effects. So in short, we built an integrated pipeline that one reveals a novel endothelial to keratinocyte intermediate cell state in AD. Two, identifies both endogenous and deep learning generated microRNAs predicted to regulate the state. And finally, proposes a scalable framework for precision RNA-based therapy development in skin disease. So the next few steps in this research is to functionally test these microRNAs in co-culture systems of endothelial and keratinocyte cells, and also in AD mouse models. We also want to map the signaling pathways in detail, optimize RNA delivery systems, and eventually explore clinical translation in patient samples. So this project brings together my personal experience with eczema, cutting-edge single-cell genomics, and AI-driven RNA design. My hope is that this work moves us closer to more precise, lasting treatments for people with AD, just like me, and while simultaneously opening the door for similar strategies in other inflammatory and barrier-related diseases. And before I end it off, I have a couple of thank yous. I want to thank Allison Key for her guidance, mentorship, and constant encouragement throughout this project. I want to thank the Student Research Institute for their support in making research more accessible. And finally, I want to thank you for listening to my presentation and expressing interest in my story and my work. Thank you.
Awesome. Thanks, Leon. Now we'll pass it over to what I believe is our last presentation before the break, which will be Alyssa, who will tell us a little bit about sporting environments for women. Take it away.
Hi, my name is Alyssa Yuan, and I'm a rising junior at Branksome Hall. For this project, I worked together with Bailey McLagan to investigate the existing problem of gender disparities in sporting environments and how it critically impacts the well-being of girls due to forced dropout. This is the problem at hand. It's an ongoing crisis that has been constantly overlooked and normalized, regardless of the hard evidence that girls drop out of sport twice the rate of their boy counterparts, and only 1 in 3 girls feel they belong in sport compared to 6 in 10 boys. This resulted dropout from sport doesn't only take a hobby away from girls, but it also poses detrimental consequences to their physical, mental, and social health. This is commonly rooted in historical stereotypes that girls are more fragile, they're less committed, too emotional, and only have conditional participation. But the real underlying causes have been overlooked and blamed on these stereotypes. So this is what we did in the review. We demonstrated how each of these factors, including negative body image, mistrust in support systems, inadequate resources, gendered curriculum, and non-inclusive uniforms, have a big influence on the engagement of girls with sports. We developed a framework towards how each of these factors interact with each other to exacerbate the problem at hand. And finally, we propose solutions to encourage change to limit gender disparities and promote considerations towards every participant regardless of gender. Now, these are the factors. First and foremost, negative body image is an existing stereotypical ideal body figure that drives girls to attempt to achieve this ideal physique instead of having the motivation to improve and continue in their sport to avoid being seen as masculine. Athletic sports also induce body image pressures, leading to diseases like REDS and eating disorders that induce unhealthy behaviors and forcing the athlete to drop out of sport due to overall health. Furthermore, mistrust in support systems is the lack of trust in the professional athlete relationship due to feelings of power difference and neglect. If athletes are feeling neglected or harassed, they're discouraged from reaching out about injuries or seeking proper care, allowing issues to worsen over time. And this forces them to drop out due to emotional trauma or injury. Moreover, inadequate resources is the lack of resources to perform fully and comfortably in sport, including training facilities, sports bras, and period hygiene products. These are essential for promoting body confidence, comfort, and avoiding self-consciousness. Without access to these resources, there is a lack of support to continue participating. Next, gendered curriculum is a school curriculum focusing on male-dominated sports and values instead of including variable sports and activities. This fosters feelings of exclusion and inadequacy. Since girls are treated differently from boys, it further contributes to the perceived competence, leaving a lasting negative impression and it impacts their future in sports. And finally, non-inclusive uniforms is a gendered clothing in sport that is revealing and restrictive, affecting performance in girls as it reduces comfort and confidence. This strips away girls from freedom of choice due to strict rules and serious consequences when not followed. Overall, the overarching theme is that they decrease sense of belonging, create discomfort, reduce motivation, and undermine confidence. As shown in Figure 1 at the bottom of the slide, we developed a framework and how all of these factors are interconnected through these themes: sexualization of bodies, a social narrative of athletic inferiority, and biased treatment towards boys. For example, uniform and body image are connected because since uniforms are more revealing, the body is more on display, inducing feelings of self-consciousness. And the other factors are connected through these themes as well. So why does any of this matter? This matters because of the large impact participation has on the well-being of a person. It promotes body appreciation, as sport is found to help mitigate the negative impact of developing unrealistic body standards. It plays a crucial role in social development by promoting peer interaction and social engagement. And finally, it decreases risk for mental and metabolic diseases. Overall, it plays a pivotal role of a person's health as it maintains a well-rounded well-being. So what do we do to disrupt this pattern? Coaches, professionals, and policymakers need to provide a more inclusive and safe sporting environment, voice underrepresented voices in decision and policy making, and conduct more research on dropout rates of transgender and gender nonconforming adolescents to build support systems. Now, to conclude my presentation, I would like to thank my mentor, Bailey, for supporting me and guiding me throughout this experience, and for the Harvard undergraduate SRI Open Bio Research Program for providing me this valuable opportunity to gain experience in research. Thank you.
All right, awesome. With that, I think that's all of our presentations before our break. So I'm taking a look at the schedule here, and it seems like we are around like five-ish, 6 minutes ahead of schedule. So this break will be a lot longer than anticipated. The first presentation, which is Ria's right after the break, is going to be at 1:36 p.m. So let's try to have everyone back here by 1:34 p.m. Eastern time. So that's exactly 10 minutes from now. So you can feel free to turn your cameras off, stretch, go get water, watch TV, play Clash of Clans, do whatever you want to do. Just be back in 10 minutes at 1:34, and then we'll start the presentation at 1:36 for the second half of the symposium. Great job so far. You guys are killing it. Let me know if there's any questions. All right, guys, let's start coming back. I'll give folks a minute or two to trickle back in. In 1 minute, we're going to start up with Ria's presentation, and then we'll go from there. If you're one of the people that has your camera off, please come back from the break, turn your cameras on. We want to resume the symposium, and of course, all of you need to have your cameras on for the last part, um, as we're watching some of the other mentees present. So please be respectful and keep your cameras on if you're a mentee. If you're a mentor, you're welcome to keep your camera off or turn it on if you're able to. All right, it is 1:36. We're going to go ahead and kick it off, kick the second half of our symposium off with Ria, who's going to tell us a little bit about the bridge within the brain. Take it away.
Thank you so much. Good afternoon. My name is Ria Govani, and today I'll be presenting our research project, a systematic review of the bridge beneath the brain, discussing astrocyte envy, calcium microdomains, and neurovascular coupling. This project was completed in collaboration with Aaron Santos from the Oregon Health and Science University, whose contributions were integral to this work. Now we'll begin with the abstract and introduction portion in the top left corner. Astrocytes, the star-shaped glial cells of the central nervous system, are often thought of as passive support cells. In reality, they are active conductors translating neuronal commands into vascular changes that regulate blood flow and protect the blood-brain barrier, or BBB. Our work focuses on calcium microdomains and astrocyte envy, rapid localized calcium signals that act as control hubs for both the BBB integrity and neurovascular coupling, which is the linking of neuronal activity to changes in blood flow, where blood vessels, as a result, adjust their diameter to deliver the right amount of oxygen and nutrients exactly where and when they're needed. These calcium signals travel through two key channels, TRPV4 and TRPV1. Overall, the goal of this project was to systematically review and synthesize how microdomains influence vascular function and how this knowledge could lead to therapies for neurological diseases such as Alzheimer's, epilepsy, and stroke. Moving on to our method section in the bottom left, we followed PRISMA 2020 systematic review guidelines, screening over 240 records related to and narrowing to 53 primary research studies. These studies used rodent and human models, calcium imaging, and molecular manipulations targeting channels like TRPV4 and TRPA1. We also performed a simplified Robbins eye bias assessment, which is the colorful table at the bottom of the methods section, evaluating study design, confounding factors, and reporting transparency. And with that foundation in place, let's move on to our results and discussion, where we synthesize over a decade of experimental work to map the architecture, functions, and therapeutic potential of these incredible microdomains. First of all, you may be wondering why we chose the bridge metaphor for this project. If we direct ourselves to the role of the astrocyte section, the neuron serves as the center of control, and the astrocyte serves as the signal bridge, and cars traveling this bridge serve as calcium signals, while the blood vessels ultimately serve as a gate, which opens wider as the traffic of cars increase, showing the relationship between neurons and astrocyte and for calcium signaling, and the dilation of blood vessels within the brain. If we move on to our architecture and dynamics of microdomain section, we investigate the architecture and dynamics of end-foot microdomains. Here, astrocyte envy generates fast, localized calcium transients organized by TRP channels, mitochondria, and ER junctions. These microdomains are not random, and they are precisely organized in space and time, allowing astrocytes to translate neuronal activity into vascular responses. In calcium's role in BBB integrity, TRPV4 and TRPA1 channels maintain tight junction stability in BBB. Disrupted calcium balance seen in normal brain aging, epilepsy, and Alzheimer's compromises barrier function. This research proved that preserving calcium homeostasis may slow neurodegeneration and improve drug delivery into the brain. In the calcium control of neurovascular coupling section, Astrocyte microdomains also fine-tune blood flow, adjusting vessel diameter in response to neuronal activity. They don't just passively relay signals, they amplify and coordinate them, ensuring that blood flow matches the brain's moment-to-moment metabolic needs. In the translation of just a function to therapy section, many therapeutic strategies are emerging as a result of this research. Within our translating dysfunction into therapy section on the top left, we can see that TRPV4 inhibition has also been shown to reduce seizures and even preserve microdomain function in vivo. Astrocyte-targeted nanoparticles could deliver drugs directly to these control hubs, restoring vascular regulation without systemic side effects. This approach could be incredibly transformative for diseases like epilepsy, Alzheimer's, and stroke. Next generation experiments and tools. The next steps involve super resolution imaging and live cell nanosensors in order to map calcium in unprecedented detail, while optogenetics will allow us to control specific calcium channels and improve causality. And BBB on the chip platforms will translate these findings into human relevant systems without experiencing the limitations of strict code of ethics enforced on human testing and limited funding for expensive in vivo experimentation. Understanding this bridge within the brain could bridge the gap between theoretical research and practical therapies, paving the way for a new generation of less invasive treatments for many neurological diseases. I would like to thank Aaron Santos for being so engaged in this research and being an incredible source of inspiration in this project. We, of course, would like to thank the Harvard Student Research Institute at OpenBio for their support in making this research possible. Thank you.
Awesome. Thank you, Ria, and great presentation. We're now going to move on to Luke, who's going to tell us a little bit about antibiotic monotherapy.
So, hi everybody. Thank you all so much for being here today. Today I'll be presenting my systematic review and meta-analysis mentored by Calvin Trin titled The Impacts of Topical Antibiotic Monotherapy and Combination Therapy on Atopic Dermatitis Severity, a systematic review and meta-analysis. So first, I just want to start off with a question. When you meet someone initially, what's the first thing you typically notice about them? Because for me, it's their appearance. And for people living with atopic dermatitis, that means visible redness, scaling, inflammation, etc. So AD, also known as eczema, is a chronic inflammatory skin condition that affects up to 20% of children and 10% of adults worldwide. One of the main drivers of AD flares is the bacterium Staphylococcus aureus, which often overgrows on the skin during flare-ups. And in order to manage these flares, clinicians often prescribe topical antibiotics, which is often used alongside corticosteroids to reduce inflammation. However, while antibiotics do reduce infections and therefore improve symptoms short term, they can also disrupt the skin microbiome and the healthy microbes that come with it, which is known as dysbiosis, thus contributing to antibiotic resistance as well. So, the objective of our study is to investigate this balance of potential benefit and risk in using topical antibiotics in eczema. So the ultimate overarching question of our topic was, do topical antibiotics, either alone or in combination with other treatments, improve AD severity, and what risks do they pose? So we examined both the effectiveness and safety of these treatments with a particular focus on microbiome changes and the development of antibiotic resistance. Regarding the methods, we performed a systematic review and meta-analysis, and we searched three primary databases, namely PubMed, Central, and Web of Science. And we also applied the inclusion criteria listed below. So in total, the systematic review encompasses 12 studies evaluating both monotherapy and combination therapy. And then the meta-analysis focuses on 9 studies evaluating solely combination therapy. So we also assessed the risk of bias using the ROB2 tool, and we excluded one study for high risk of bias. And then the Prisma flow diagram below shows the selection process we used. We measured changes in AD severity, microbiome composition, and antibiotic resistance primarily, and then we calculated the pooled risk ratios with 95% confidence intervals, which we visualized in a forest plot. Regarding the methods, the meta-analysis showed that combination therapy generally did improve AD severity, yielding a pooled risk ratio of 0.79 and a confidence interval from 0.73 to 0.87, which does indicate a statistically significant effect. So, in other words, patients receiving combination therapy were more likely to improve than those without antibiotics. And we also found that combination therapy was generally more effective than monotherapy in improving AD severity. So, however, although there were some benefits reported, there were also some concerns. So, five studies did report increased antibiotic resistance, most commonly with the repeated use of fusidic acid and mupirocin, mainly because those are the two most commonly prescribed topical antibiotics. And in infants, mupirocin combined with corticosteroids did delay the restoration of the Streptococcus species on the skin, which is really negative because that's one of the beneficial microbes found in the skin microbiome. And then, moreover, one study found that adding tetracycline to corticosteroids did provide no clinical benefit, even though it was successful in eradicating the bacteria. So, in summary, topical antibiotics do provide short-term benefits for patients with AD, especially used in combination with corticosteroids during flares. However, the risks, particularly antibiotic resistance and microbiome disruption, do become more significant with prolonged or repeated use. So, based on these findings, our clinical takeaway is that topical antibiotics should be used on an as needed basis and only reserved for targeted short term interventions rather than routine treatment. So, regarding the gaps in the research, some studies did have short follow-up periods and limited microbiome data, which makes it harder to assess the long-term consequences of these treatments. And future research should elucidate the long-term impacts on microbiome recovery and resistance as well. So, thank you.
Awesome. Great presentation, Luke, and thanks for the chat. We're now going to move on to Vivian, who's going to tell us a little bit about adenocarcinoma.
Hello, my name is Vivian Zhao, and today I will be discussing the role of stress-related biomolecules in pancreatic ductal adenocarcinoma, also known as PDAC, which is one of the most deadly cancers with a survival rate of less than 15%. Now, you may be wondering, what is the connection between stress and cancer progression? Stress triggers neurotransmitter and hormone release to regulate homeostasis, but in excess can promote cancer via angiogenesis and immunosuppression. It may seem strange that a central nervous system response can affect an organ as distant as the pancreas, but a key pathology in PDAC is perineural invasion, where cancer cells invade and manipulate local neurons to secrete biomolecules and promote cancer aggression. Current PDAC treatments are limited, meaning we need to consider out-of-the-box solutions. Therefore, my literature review addresses the relationship between stress-related biomolecules and PDAC and the relevance of targeting these molecules to increase PDAC survival. I reviewed 130 PubMed articles, of which 77 reviews, research studies, and clinical cases were used to identify three main takeaways. First is that aberrant signaling of stress-related biomolecules in the sympathetic and parasympathetic nervous systems interact with the tumor microenvironment to drive cancer growth, as can be visualized in the figure on my poster. Catecholamines such as epinephrine and norepinephrine primarily increase angiogenesis, but can also promote cancer growth through epithelial mesenchymal transition and increased chemotherapy resistance. Unlike other neurotransmitters, studies show that neurotransmitters like dopamine and acetylcholine had mixed effects depending on which dopamine receptor or muscarinic receptor they bind to. This demonstrates that stress-related biomolecules could be something to target to decrease tumorigenesis and drive positive patient outcomes. So to determine if existing medications may be useful against PDAC, I discovered my second takeaway, where dysregulated neurotransmitters can contribute to neurological comorbidities in PDAC. Anxiety and depression, for example, are highly prevalent in PDAC patients with a 7 times higher rate than the general populace, and treatment is essential since these disorders can reduce chemotherapy compliance and lower patient survival. One major shared mechanism is serotonin. In depression, the depletion of serotonin is directly correlated with the depressive symptoms, while in PDAC, serotonin is essential for something called acinar-to-ductal metaplasia, which is how normal pancreas cells become cancer cells. Neurodegenerative disorders, on the other hand, like Alzheimer's disease and Parkinson's disease, have an inverse relationship. An important observation was that those who are high risk for Alzheimer's disease but not officially diagnosed had a statistically higher chance of developing pancreatic cancer. Because psychological disorders ranging from depression to Alzheimer's function through similar mechanisms to PDAC, these medications may be repurposed to combat pancreatic cancer. Therefore, my last takeaway is that many neuropsychiatric medications are promising for treating PDAC through drug repositioning. Two main groups successfully repurposed against PDAC are antipsychotics and benzodiazepines. Antipsychotics, typically prescribed for psychosis, consistently have had high success in PDAC, especially when combined with chemotherapy. In particular, two combinations, aripiprazole and cisplatin, and pimavanserin and gemcitabine both amplified the anti-tumor abilities of the chemotherapeutic drugs. Interestingly, there were a few Parkinson's drugs like carbidopa and pimavanserin that still had chemotherapeutic effects despite PDAC's inverse relationship with Parkinson's disease. Meanwhile, benzodiazepine success depended on activating OGR1, a G-coupled protein receptor that can increase other oncogenetic proteins. And unsubstituted benzodiazepines that activate OGR1 have many immunosuppressive side effects, such as increased desmoplastic stroma and inflammatory signaling. Meanwhile, and substituted benzodiazepines that did not increase OGR1 were successful in reducing tumor area and cyclin proteins. These drugs may not only be able to serve as an unorthodox cancer treatment, but can also treat other cancer-associated symptoms like insomnia and anxiety. Other drug classes such as cholinesterase inhibitors, NMDAR antagonists, and selective serotonin reuptake inhibitors also have anti-cancer effects, which can be seen in the table on my poster. Therefore, by confirming the role of cancer reprogrammed neurotransmitters and comorbidity between PDAC and neurological disorders, research also supports neuropsychiatric drug repurposing in combination therapies with traditional chemotherapeutic agents. We often forget how important mental health is, and in the context of chemotherapy, it can make a critical difference in patient survival. Finally, I would like to acknowledge SR staff for organizing this amazing program and my mentor, Brielle, who was very dedicated to helping me and my project. Thank you for listening.
Awesome presentation, Vivian. Really appreciate it. We're now going to move on to Aislinn, who's going to tell us a little bit about regulatory RNA.
Hi everyone, my name is Aislinn and I performed a review on how Staphylococcus aureus adapt to iron limitation in the host, focusing on the role of a small regulatory RNA called ISRR. S. aureus is a gram-positive bacterium that normally lives harmlessly on the skin and mucosa, but it can become pathogenic when it enters deep tissues or the bloodstream. It causes a range of diseases that range from mild skin infections to life-threatening pneumonia or septicemia. As shown in Figure 1, under background at the center left of my poster, S. aureus is the leading cause of death among non-tuberculosis bacterial infections worldwide, with over 1 million deaths in 2019. One major host defense S. aureus must overcome is nutritional immunity, the sequestration of essential metals to restrict bacterial growth. In Figure 3, under the bacterial response to iron limitation on the upper right corner, illustrates this process, showing how proteins like transferrin, lactoferrin, and ferritin bind iron, while neutrophils secrete calprotectin to chelate iron, zinc, and manganese at infection sites. This creates an iron-starved environment that pathogens must adapt to in order to maintain their survival and virulence. To survive these conditions, S. aureus uses several acquisition systems. It produces cytophores such as staphyloferrin A and B to capture iron and from host proteins. It also uses the ISD system to extract heme from hemoglobin and employs staphylophine to gather other metals. Figure 4 at the bottom left under the same section summarizes these strategies, including ABC transporters that were used for SATR4 uptake and the ISD system. Moving on, the iron uptake must be tightly regulated to prevent toxicity. The ferric uptake regulator FER senses intracellular iron levels and adjusts gene expression accordingly. Figure 5 under the first section at the very top, it shows that in iron-rich conditions, FER binds Fe2+ and represses both iron acquisition genes and ISRR. So basically, when iron is scarce, FER is inactivated, lifting this repression and allowing iron uptake and ISRR expression. And ISRR acts post-transcriptionally to repress the translation of non-essential iron-heavy enzymes, particularly in the TCA cycle as well. As depicted in Figure 6 at the very bottom of that same first section, this repression conserves iron for essential processes and shifts metabolic priorities under iron-limited conditions. Moving forward to my next section, ISRR. It is a small FER-regulated RNA that plays a central role in the iron-sparing response of S. aureus. Once it's expressed, it uses 3 conserved C-rich regions to base pair with target mRNAs, usually near the ribosome binding site, and blocking translation without typically degrading the mRNA. And this allows the bacterium to quickly reverse repression if conditions change. So most of ISRR's validated targets, which I summarized in Table 1 in green, are enzymes that use iron-containing cofactors but are not essential for survival under iron-limited conditions. Many of these are involved in the TCA cycle, such as aconitase, succinate dehydrogenase, and citrate synthase. Others are linked to oxidative stress defense, anaerobic respiration, and amino acid metabolism. A good example is aconitase, shown in Figure 9 at the very bottom of that ISRR section. ISRR binds directly to the ribosome binding site and physically blocking the translation and reducing TCA cycle activity, as I just mentioned. And this conserves iron by preventing the synthesis of large iron-sulfur clusters containing proteins when the metal is scarce. And by doing this across multiple pathways, ISRR helps redirect iron towards processes that are absolutely required for the growth and survival. Pathogenesis studies show that ISRR's role in metabolism directly impacts the ability of S. aureus to cause disease as well. In mouse pneumonia models, strains lacking ISRR had significantly lower colonization in both lung tissue and bronchoalveolar fluid compared to wild type. And in septicemia models, mice infected with the deletion strain survived actually much longer, and this demonstrated virulence. So restoring ISRR in these mutants restored pathogenicity to wild type levels, confirming that the phenotype was specific to the loss of ISRR. Mechanistically, ISRR appears to influence virulence indirectly by repressing iron-heavy metabolic enzymes. It alters metabolic pools sensed by regulators such as CODI, and then modulate the SAERS two-component system and downstream toxin production. So in summary, the FER ISRR network enables S. aureus to adapt to iron limitation by conserving iron for essential processes while indirectly influencing virulence. Future work should aim to define the complete ISRR target set under diverse infection conditions, distinguish direct from indirect regulation, and explore ISRR as a potential antimicrobial target. I would like to thank my mentor, Gustavo Rios Delgado, for his support throughout this project, as well as the Student Research Institute program for providing the opportunity and resources to conduct this research. And once again, thank you all for listening to my presentation.
Thank you for an amazing presentation. Really appreciated it. We are now going to go to a recording. Let's see who this is. So this is Ari, who unfortunately wasn't able to make it, but she'll be recording, or she'll be presenting via recording on her research on reactive oxygen species. Let me know if you can hear this audio, and if not, we'll troubleshoot a bit.
Good afternoon. My name is Ari, and today I'll be presenting findings from a recent systematic review titled The Role of Reactive Oxygen Species in Mitochondrial Impairment in Alzheimer's Disease Progression, Insights from Animal Studies. So to start us off, Alzheimer's disease, or AD, is a progressive neurodegenerative disease that affects over 7 million Americans with no known cure or proven method of prevention. Despite decades of research, the exact causes of the disease remain unclear. However, growing evidence points to oxidative stress as a major driver of disease progression. Oxidative stress occurs when cells produce too many reactive oxygen species, or ROS, and can't neutralize them with antioxidants. This imbalance damages proteins, lipids, and DNA, especially in the brain, where neurons rely heavily on mitochondria for energy. Our goal was to systematically review in vivo animal studies published since 2015 to identify the major causes of oxidative stress in Alzheimer's models and highlight emerging patterns. We searched PubMed using specific mesh terms for Alzheimer's disease, reactive oxygen species, and mitochondrial dysfunction. Out of 361 articles, 28 original research studies met our inclusion criteria. We only included in vivo animal models and excluded reviews, in vitro studies, and non-English texts. Each article was assessed using Cycle's risk of bias tool to ensure quality. So as you can see in the image, the main findings fall into 6 major categories. Number 1, pathological proteins show that mutations in genes such as APP and PS1 drive amyloid beta production, which accumulates in mitochondria and drives ROS production. These proteins also impair electron transport train, causing mitochondrial dysfunction across mice, rats, flies, and other animals. Number 2, mitochondrial quality control. Disrupted mitophagy, the process of removing damaged mitochondria, leads to ROS accumulation, impaired PINK1-PARKIN pathways, and proteins such as TUFM and MIRO1 were frequently cited as regulators of this dysfunction. Number 3, environmental factors. Exposure to metals such as copper, iron, lead, and mercury, as well as pesticides like paraquat, significantly increased ROS and worsened cognitive symptoms in AD models. We found that these agents also interacted with amyloid beta pathology to further induce oxidative stress. Number 4, redox signaling regulators. The downregulation of antioxidant regulators such as NRF2, TXN2, and TUFM, and the upregulation of ROS generating enzymes such as NOx4 contributed to redox imbalance and cell death through mechanisms such as ferroptosis. Number 5, metabolic and endocrine stressors, such as cholesterol overload, disrupted insulin-like growth factor signaling, and AGE-rich diets all impaired mitochondrial mechanism and increased ROS. Interestingly, even muscle disuse elevated oxidative stress in Alzheimer's models. And last but not least, bioenergetics dysfunction. Defects in oxidative phosphorylation, especially in models with ApoE4 or PSEN1 mutations, caused mitochondrial calcium overload, electron leakage, and ROS bursts. One study even found that reverse electron transfer, a complex sponge, drove ROS in aging fruit flies. So together, these findings show that oxidative stress is not a secondary symptom, but a central mechanism in Alzheimer's disease progression. It results from a complex interplay of genetic mutations, environmental exposures, metabolic disruption, and mitochondrial failure. Although the study is limited to animal models, many of these mechanisms are conserved in humans. Our results support the idea that targeting oxidative stress, particularly through antioxidant therapy and mitochondrial repair, may be a promising strategy for slowing or preventing Alzheimer's disease. Future work should validate these insights in human models and explore therapeutic approaches that address oxidative damage early in disease development. Thank you for your time. I hope this synthesis offers a broader framework for thinking about the role of oxidative stress in Alzheimer's. Lastly, I'd like to thank my mentor and SRI for this wonderful opportunity and the guidance they've provided.
Awesome. All right. Thanks to Ari, who presented virtually. We are now going to move on. Now going to move on to Arush, who's going to tell us a little bit about PCB technologies. Take it away.
Thank you so much for the introduction. So, as you all know, there is a growing problem of pollution on our planet today, with an estimated 50 billion tons of pollution across our planet. And this figure continues to grow by over 160 million tons each year. Now, a large part of the global pollution problem is the global e-waste industry. As countries develop, there is a growing amount of phones and computers and personal devices, and when these are thrown away, they contribute to the global e-waste, which is an estimated 62 million tons of plastic pollution every single year. Now, a large portion of this problem comes from printed circuit boards. Circuit boards, as you may know, are used in a majority of personal devices. And so when every single device is thrown away, a circuit board is thrown away with it. Now, there are three key things in the printed circuit board industry which lead to the pollution that it is causing. The first is the use of toxic heavy metals in these printed circuit boards. Metals such as mercury, cadmium, and lead are used in these printed circuit boards, and so when they're thrown away, they leach into the environment and cause a pollution in the environment of toxic heavy metals. Additionally, the use of subtractive manufacturing in the printed circuit board industry, in which the whole is broken apart to make the printed circuit board, causes a lot of waste and causes a lot of emissions in the printed circuit board industry. Lastly, the use of substrates such as flame retardant 4 and flame retardant 2 in printed circuit boards are not biodegradable at all. And so what this does is it causes a buildup of printed circuit boards in which they are not naturally degraded. And so this also contributes to the global pollution problem. Now, my project focused on a literature review using sources across the internet, and the goal was to emphasize the developments in the printed circuit board industry and the future objectives in this industry in order to maintain and improve the sustainability of printed circuit boards. So before we go on to the present, I would like to talk about the historical perspectives on printed circuit boards. So printed circuit boards were developed around 100 years ago. And since then, they've been using these toxic heavy metals and subtractive manufacturing and these poor substrates in their design. And so for over 100 years, we've been integrating these poor practices into the industry of printed circuit boards as a whole. Now, there have been a few current solutions to some of these problems, the first of which came in the form of additive manufacturing. So subtractive manufacturing, as I mentioned before, was when the whole was broken down into separate parts and there was a lot of waste as a result. In additive manufacturing, each layer of the printed circuit board is assembled individually, and then these layers are stacked upon each other to reduce the waste produced. And in fact, emissions have been reduced significantly with additive manufacturing, reducing the estimated emissions by over 50% compared to subtractive manufacturing practices. Additionally, there has been a use of recyclable materials in these printed circuit boards recently. And so we're seeing the use of different substrates, such as PET, polyethylene terephthalate. And so what this thermoplastic does is it's a recyclable material. And so that increases the circularity in the printed circuit board industry. We've also seen the use of degradable plastics in the printed circuit board industry. So we've seen the incorporation of biopolymers such as flax fibers and jute fibers and cellulose fibers into printed circuit boards, which increases their biodegradability. And we've also seen an optimization in the soldering process. So in the soldering of printed circuit boards historically, there was a lot of waste. And so by using AI optimized workflows in very recent years, we've also optimized the soldering in printed circuit boards. Now, there are a lot of applications of these technologies in printed circuit boards beyond just the environmental impact, which incentivizes other industries to use these improved circuit boards. And these include biomedical electronics, in which dissolvable circuit boards can be made, and so they can dissolve naturally in the body. Environmental modeling and monitoring, in which we can use printed circuit boards which can be biodegraded in the environment without having a need to pick back up the circuit board, and secure electronics, in which we can develop self-destructing printed circuit boards as well. And so beyond just the environmental uses, there's also an industry incentive to use these improved printed circuit boards. However, there is still a need for certain improvements in the sustainability of printed circuit boards. And so the most pressing of these issues is the space. So these improved, more sustainable printed circuit boards take up nearly 20% more space compared to traditional printed circuit boards. And so this reduces their use in electronics currently. Additionally, there is a lack of thermal tolerance and moisture tolerance in these sustainable printed circuit boards. For example, the flax, jute, and cellulose fibers mentioned previously pick up water a lot more efficiently than traditional printed circuit boards, and that threatens their usage in personal devices. And so these issues require future work in the printed circuit board industry. And so the ultimate goal of the printed circuit board industry would be to reach a state where they reduce emissions even more and where they become even more biodegradable, improving their sustainability and circularity in the current economy. I would like to thank my mentor, Ms. Swami of Northeastern University, and I would also like to thank SRI and the Harvard Undergraduate Open Bio Laboratory for making this project possible. Thank you.
Awesome. Great presentation, Arush. Love to see it. We're now going to move on to Harmrit, who's going to talk to us a little bit about Parkinson's disease detection.
Good afternoon. My name is Harmer Bol, and today I'll be presenting our work on a new method for detecting Parkinson's disease, the multimodal fusion of 3D SPECT and clinical data using cross-attention. So Parkinson's disease is a debilitating neurodegenerative disorder whose diagnosis is primarily clinical, relying on a neurologist's evaluation. This process can be highly subjective, leading to a misdiagnosis rate as high as 25%. This is especially true when differentiating PD from other Parkinsonian syndromes like multiple system atrophy or progressive supranuclear palsy, which can present with similar symptoms but have different prognoses and treatments. This diagnostic uncertainty can cause significant distress or for patients and delay critical treatment. While neuroimaging tools like DAT-SPECT scans are powerful for assessing the dopamine pathway affected in PD, their visual interpretation can also be very subjective. This highlights a clear need for more objective and reliable diagnostic aids. So to address this challenge, we moved beyond analyzing a single data type. Our research introduced a novel multimodal deep learning framework that uses two key sources of information: the raw 3D SPECT brain images and a corresponding set of 12 clinical and quantitative parameters, such as the spatial binding ratio. And you can see that in figure 1 on our poster. To address this, traditional models often combine data using late fusion, basically just sticking the features together at the end. This can fail to capture the complex underlying relationships between the two different data types. So our approach, inspired by the CLIP model, uses constructive learning and cross-attention to force the model to align the embeddings from both the image and the tabular data. Our 3D CNN encoder learns spatial features from the raw image volume, while an MLP encoder processes the high-level quantitative metrics. The cross-attention mechanism then acts as a bridge, allowing each branch to weigh the importance of features from the other, creating a truly integrated representation. So, how did our approach perform? On our held-out test set of 103 subjects, our 3D multimodal achieved an excellent accuracy of 95.1% and an area under the curve of 0.984. And you can see in the ROC curve in figure 2. So the confusion metric shows the model correctly identified 48 out of 51 Parkinson's patients and 50 out of 52 healthy controls with very few errors. Our ablation study also proved the value of this fusion. Models using only image data or only tabular data performed very poorly, with accuracies under 80%. Our multimodal deep learning approach represents a significant leap in performance. Next, we can see in figure 3 our TSNE plot, and this plot explains why our model works so well. On the left and in the center, you can see that using a single data type fails to separate the healthy and Parkinson's patients. But on the right, our fused representation shows a near-perfect linear separation between the two classes. This visually confirms our model is learning a highly discriminative feature space. So our results demonstrate that contrastive alignment is a highly effective strategy for integrating neuroimaging and clinical data. The significance of this result is twofold. First, it confirms that the 3D spatial information offers a clear advantage over 2D approaches, as seen in our ablation study. Secondly, and more importantly, the success of diffusion shows that the model is learning a representation that is more robust than the other data type alone. This suggests a potential to reduce the diagnostic uncertainty that arises from relying on a single, sometimes ambiguous, source of information, which directly addresses the core clinical challenge we set out to solve. While the study does have limitations, such as dataset size and focus on binary classification, it serves as a strong proof of concept. Future work will involve validating this framework on larger multi-class datasets to differentiate between various Parkinsonian syndromes. In conclusion, this multimodal framework is a promising step towards developing more objective and reliable computer-aided tools. By effectively fusing data, we can move closer to reducing diagnostic uncertainty for patients with neurodegenerative diseases. Thank you for your attention. I would like to thank my mentor, Jinan Deng, and Harvard Open Bio Undergraduate Laboratory. Thank you.
Awesome. All right. Thanks, Harmrit, for a great presentation. We're now going to move to Roshni, who's going to talk to us a little bit about iPSCs in cardiomyocyte regeneration. Take it away.
Hi, my name is Roshni Ball, and today I will be talking about my literature review on rewiring the ischemic myocardium. So, some background information first. Myocardial ischemia happens when blood flow to the heart muscle is reduced or blocked, most often because of coronary artery disease. That drop in blood flow means the heart muscle isn't getting enough oxygen, and over time it leads to the death of cardiomyocytes. The problem is, instead of being replaced with new functioning heart muscle, that area gets filled with fibrotic scar tissue. And scar tissue cannot contract, it can transmit electrical signals properly, and it essentially weakens the heart over time, increasing the risk of heart failure and arrhythmia. Now, the human heart doesn't really have the capacity to regenerate these lost cells on its own. This is why regenerative medicine, and specifically stem cell therapy, has become such an active area of research. In this literature review, we focused on the comparison between mesenchymal stem cells, or MSCs, and specifically a subpopulation called mu cells, versus induced pluripotent stem cells, or iPSCs. The potential of stem cells can be leveraged particularly through a mechanism of paracrine signaling, which is actually where most of the benefit of stem cell therapy can come from. In this sense, what we know now is that most of the therapeutic effect comes from the cells releasing signaling molecules or bioactive molecules, such as growth factors, cytokines, and extracellular vesicles. These molecules then act on the surrounding tissue to promote new blood vessel formation, prevent further cell death, and modulate inflammation. So for mu cells, these are a small fraction of MSCs marked by a surface protein called SSEA3. They're pluripotent-like, they can survive under stress, and they're naturally immunotolerant because they express a certain encoded protein called HLA-G. After IV infusion, they can detect chemical gradients from the injured myocardium, and specifically through sphingosine 1-phosphate signaling, and also migrate directly to the damage. In other words, their homing ability, which is amongst their biggest advantages. Once they arrive, they secrete a potent mix of reparative molecules including VEGF, HGF, and enzymes that remodel scar tissue. In preclinical studies, giving mu cells intravenously has reduced infarct size by roughly 50% and improved left ventricular function without causing arrhythmias or any other adverse effects. On the other hand, iPSCs are made by taking a normal adult cell, like a skin fibroblast, and reprogramming it into a pluripotent state using specific transcription factors such as OCT4, SOX2, etc. These iPSCs can then be differentiated into cardiomyocytes, called iPSC-CMs, or into mesenchymal-like cells. Their secretome, or the collection of molecules they release, contains cardioprotective microRNAs, like microRNA 21 and 210, and several cytokines. These molecules can activate autophagy, reduce fibrosis, and promote new blood vessel growth. In animal models, direct injection of iPSCMs or their exomes into the heart has improved ejection fraction by about 10 to 15% and reduced fibrosis in the damaged area. The main limitations are that iPSCs don't naturally home to injury sites, so they have to be delivered directly into the myocardium. But then they run the risk that any residual cells can form teratomas. So when you put these two approaches side by side, both are clearly capable of improving cardiac outcomes in preclinical models. And in both cases, the main driver is paracrine signaling, rather than the long-term survival of the translated cells themselves. They each have their own advantages and limitations, but for now, it's seen that mu cells offer the benefit of a simple IV delivery, natural homing, and have a very low risk of adverse side effects. But ultimately, the main difference is the two really different ways of approaching the same goal, given that one uses cells that are already tuned by nature to find and repair injury, whilst the other uses cells that can be engineered and customized for precision applications. As the population ages and ischemic heart disease becomes even more prevalent, it's likely that the future of cardiac regeneration will less likely favor choosing one over the other, but rather demand finding ways to combine their strengths into a unified therapeutic strategy. For instance, the use of iPSCs can be explored in combination with the emerging field of personalized medicine, as an example. And that's the direction that could take us from treating the damage to truly reversing it. I would also like to thank the Harvard SRI Open Bio team and my mentor, Milan Dow, for helping me with this project. Thank you.
Awesome. Great presentation, Roshni. Really appreciate it. We're now going to move to Ryan, who's going to talk to us a little bit about patient outcomes for those with cochlear implants.
Hello, everyone. My name is Ryan Kumar, and for my project, I conducted a literature review of treating hearing loss with cochlear implants, or CIs. To give some background, in the bottom left corner, a CI is a surgically implanted electronic device that replaces the function of damaged parts of the cochlea, a snail-shaped fluid-filled cavity in the inner ear. Unlike hearing aids that simply amplify sound to make it louder, CIs directly stimulate the auditory nerve branching off from the cochlea with electrical signals, helping restore a sense of sound to those with severe to profound hearing loss. As seen in Figure 2, a CI contains three external components, a microphone, speech processor, and a transmitter, as well as two surgically implanted internal components, a receiver and an electrode array. The processor is an especially important component, as it analyzes sound and applies various algorithms and techniques to refine sound clarity. However, although there have been many advancements in CI technology, two large challenges remain. One, patients often struggle to understand speech and background noise in music, creating the need for the advanced sound processing evaluated in this review. And two, CI patients require plenty of postoperative care to optimize hearing improvements, motivating us to outline a structured, patient-centered method of implementing it. For my methods, I searched three academic databases using specific inclusion criteria of referencing peer-reviewed articles, systematic reviews, and clinical trials on CI use among adults published from 2010 to 2025. As seen in Figure 1 in the top left, the content was grouped into three foundational concepts: sound processing, postoperative care, and patient outcomes. So to begin at the top right, there are a few key processing techniques that help users hear more clearly in challenging and noisy acoustic environments. First, auto-sensitivity control, or ASC, automatically adjusts microphone sensitivity based on surrounding noise levels, prioritizing the capture of closer or more important speech and reducing softer or more distant noise. Second, acoustic scene classification is a central part of sound processing. It first extracts sound features like pitch or rhythm, then matches them to known sound profiles to classify the environment, and finally implements the most optimal processing tool suited for that given environment. And third, adaptive dynamic range optimization, or ADRO, continuously adjusts the gain across various ranges of frequencies in real time, directly amplifying softer sounds and reducing intolerably loud ones. Most importantly, research has shown that these three automatic responsive techniques are highly effective and have optimal hearing outcomes when combined, most clearly demonstrated by a large-scale clinical study by Wolf and colleagues. Based on the study, the Nucleus 6 processor, using all three previously mentioned techniques, resulted in significantly higher speech recognition than the Nucleus 5 processor that only used standard settings. Moving down below, we find that comprehensive auditory rehabilitation, or CAR, is the core method of postoperative care, supporting speech perception and device adaptation through four key components. First, in sensory management, an audiologist activates and programs the CI in a process known as mapping, creating a personalized sound profile. Second, in instruction, patients are guided on device operation and use and given a structured follow-up schedule beginning with device activation and including regular assessments of hearing progress and device mapping adjustments based on behavioral feedback and physiological measurements of electrical stimulation. Third, perceptual training trains the brain to interpret the signals sent by the CI as sound. It involves at-home passive listening of radio, TV, etc., and also active exercises that involve feedback and interaction, either with a clinician or using computer-based auditory training, which we find to be a highly effective method at improving speech recognition. And the fourth and final component of CAR, counseling, is done by speech-language pathologists who encourage patient engagement with auditory training and support their well-being and communication. An important consideration of CAR is early activation, turning the device on within 1 day to 1 week post-surgery, as opposed to conventional activation 3 to 4 weeks post-surgery. A multitude of studies indicate that early activation is safe, resulting in improvements in hearing equivalent to those of post-recovery activation. Although we find that minor complications like swelling or vertigo require careful management, early activation was found to be best suited for most patients because it accelerates hearing outcomes and device adaptation, increases patient satisfaction, and potentially reduces the duration and cost of post-op care. Now, in patient outcomes, proper hearing loss treatment with CIs not only improves hearing, but also supports social interactions, confidence, and overall quality of life. Moreover, in older adults, they reduce the risk of cognitive decline and potentially dementia by improving working memory, concentration, etc. So in conclusion, we find that a personalized and evidence-based approach that one, combines advanced sound processing techniques, two, implements early activation if appropriate, and three, administers consistent CAR across a structured follow-up timeline of post-op care is essential to optimizing patient outcomes and health-related quality of life with CI-based hearing loss treatment. And lastly, I would like to thank my mentor, Jae-Yoon Lee, for guiding me throughout this review, and the Harvard Undergraduate Open Bio Laboratory for making this program possible.
Awesome. Thank you, Ryan, for the amazing presentation. We're now going to move on to Jasmine, who's going to talk a little bit about effects in Alzheimer's.
Hi everyone, my name is Jasmine, and my project examines the relationship between the lung microbiome and the progression of Alzheimer's disease with a focus on the understudied pathway of the lung-brain axis. Alzheimer's disease, or AD, is one of the deadliest neurodegenerative diseases. It affects around 45 million people worldwide with limited treatments or known origin points. Its symptoms are characterized by hallmark features like beta amyloid plaques, tau tangles, and neuroinflammation. While most AD research focuses on the brain itself, my review proposes a different question. What if distant sites in the body, like the lungs, could contribute to AD development? During the COVID-19 pandemic, there was a notable spike in AD diagnoses alongside the rise in respiratory illnesses. This correlation has drawn researchers' attention to the lung-brain axis, which is a pathway for immune, microbial, and molecular communication between the lungs and the central nervous system. After scanning 170 papers and ultimately landing on 51 relevant sources consisting of case studies, primary literature, and literature reviews, my review aims to highlight the idea that lung microbiota dysbiosis, or an imbalance in the lung bacteria, could influence Alzheimer's progression in three major ways: direct bacterial transfer, indirect immune signaling, and extracellular vesicle communication. Firstly, in rare but extreme infections like bacterial meningitis or pneumonia, the blood-brain barrier, or the BBB, which usually wraps around and protects the brain, could become compromised. This allows bacteria from the lungs to enter the brain through the BBB and trigger a neuroinflammatory response, as seen in my figure 1. Infections like Streptococcus pneumoniae have been shown to trigger cytokine infiltration, activate microglia, and increase beta amyloid and tau production in the brain. This directly mimics the AD pathology, signaling that direct bacterial invasion from the lungs plays a role in the brain. But in more common situations, where bacteria doesn't cross into the brain, the lungs can still send damaging signals. Studies involving lung infections from Pseudomonas aeruginosa in mice revealed elevated levels of pro-inflammatory cytokines such as IL-1 beta, IL-6, and TNF-alpha, which are known to enhance beta amyloid processing and promote tau tangle formation, both of which are central to AD pathology. The third mechanism involves extracellular vesicles, or EVs, which are important communicators on the lung-brain axis. EVs are small lipid-bound particles released by cells during an infection. As shown in Figure 2, EVs can transport proteins, nucleic acids, lipids, and microRNAs through the BBB. In the AT context, they can carry inflammatory molecules, bacterial components, and lipids across the BBB. Once in the brain, they activate glial cells and trigger neuroinflammation, as seen in Figure 3. Studies have shown that EVs released during lung infections contain caspase-3, cytokines, and bacterial lipopolysaccharides, all of which have been implicated in worsening AD-related neurotoxicity. In addition to mapping these pathways, I also explored whether antibiotics used to treat lung infections could have secondary effects on AD symptoms. Several antibiotics, including amikacin, doxycycline, ceftriaxone, and azithromycin, have shown neuroprotective properties. For example, doxycycline reduced microglia activation and improved cognitive function in mouse models, while ceftriaxone significantly lowered beta amyloid deposition, and azithromycin showed anti-inflammatory effects. These data suggest that certain antibiotics may both clear respiratory infections and reduce AD progression. In conclusion, the lung-brain axis is a promising but under-investigated avenue in Alzheimer's research.
As summarized in my literature review, the lung microbiota, whether it be through bacteria, cytokines, or extracellular vesicles, can all influence the central nervous system in ways that closely align with AD pathology. The use of antibiotics that target lung infections could also be a new therapeutic intervention for lessening AD symptoms in the future. Though it's an emerging field, this review highlights the importance of viewing Alzheimer's not just as a brain disease, but potentially as a whole body disease, where AD could be impacted by organs that we don't normally associate with cognitive decline, such as our lungs. I would like to thank my amazing mentor, Brielle, for her guidance throughout this whole project, and the Open Bio staff for providing this wonderful research opportunity. Thank you so much for listening.
Awesome. Great presentation, Jasmine. We are now going to move on to Varnika, who's going to tell us a little bit about arsenic-affected regions in the U.S.
Hello, my name is Varnika Ravi, and I'm here to present my research in collaboration with my mentor, Kelly Tucker, about environmental and rural determinants of cancer. During the Renaissance, arsenic was given the name inheritance powder because it was commonly used to poison family members for financial gain. Fast forward to today, and people living in more than half of the United States are exposed to levels of the same poison, arsenic, above EPA limits. The difference is, this time the exposure isn't deliberate. It's coming from something as basic and essential as drinking water. Arsenic is colorless, odorless, and tasteless, making it impossible to detect without testing. With long-term exposure, it can lead to cancer, the second leading cause of death in the US. Rural communities, especially in western states, are at heightened risk because they rely on private wells that are not federally regulated. This combination of environmental hazard and healthcare inequity is a perfect backdrop for cancer disparities. That's why this study aims to 1. Identify geographic areas where cancer risk is elevated due to environmental exposure. 2. Support more targeted public health interventions in underserved areas. And 3. Inform stronger environmental regulations for water safety and rural healthcare. To do this, I focused on Arizona and California, two states with high arsenic levels and accessible cancer data sets. I gathered county-level data for 5 cancers known to be linked to arsenic exposure: bladder, thyroid, lung, skin, and kidney cancers. Arsenic data came from the National EPH Tracking Network, but because it was at a sub-county level, I calculated population-weighted county averages in order to align them with the cancer data for analysis. For the statistical analysis, I created Python-based spatial error regression models, which account for spatial autocorrelation, or the tendency for nearby areas to have similar values. I also ran Moran's 1 tests to confirm that the models accurately captured these patterns. Here's what I found. In California and Arizona, increased arsenic exposure is directly tied to higher incidence rates of 3 types of cancer: bladder, thyroid, and kidney. Bladder cancer stood out with the strongest link, having a p-value of less than 0.0001. Our model explained nearly 69% of the variation in bladder cancer rates across counties. Thyroid cancer also showed a significant association, with a p-value under 0.0001, but the model explained less variance at about 38%. When we look at rurality, bladder, lung, skin, and kidney cancers were all linked with higher rural population percentages. Skin cancer had a particularly strong rurality relationship with a p-value of less than 0.0001. However, interestingly, kidney cancer showed a negative association with rurality. The beta coefficient was negative 0.0399 and the p-value was less than 0.05. These spatial patterns aren't just statistically significant, they make sense biologically. Extensive literature has provided concrete evidence that arsenic promotes carcinogenesis through DNA hypomethylation, microRNA dysregulation, reactive oxygen species generation, and histone modification. These are all pathways that disrupt normal cell regulation and can drive tumor growth. Additionally, rurality can increase reliance on contaminated wells and reduce access to timely medical care, creating a compounded risk. Ultimately, our research shows that current EPA limits may not be enough to protect public health, especially in rural, groundwater-dependent areas. Addressing arsenic contamination and rural healthcare access together could directly reduce the more than 600,000 lives lost to cancer each year in the US. Because when something as simple as a glass of water can silently increase cancer risk, we can't afford to ignore the problem. Lastly, I'd like to thank my mentor, Kelly Tucker. Her passion for environmental science led me to explore this interesting connection between medicine and earth sciences. I also would like to extend my deepest gratitude towards the team at the SRI for this incredible opportunity. Thank you for listening.
Awesome. Thanks for the amazing presentation. We are now going to move on to Annika, who's going to tell us a little bit about taste memory analysis.
Hi, my name is Annika Foken, and I'm a rising senior at Highland Park Senior High School in St. Paul, Minnesota. My mentor is Justin Ng from the Lu Lab at Rutgers University. I'm really excited to share my work creating a brain simulation to model neural circuits and analyze taste memory. Taste isn't just a flavor. It's a gateway to neural circuits that determine feeding, metabolism, and even memory itself. By tracing the wiring of sweet and bitter pathways and their downstream connections, we can start to see how sensory experiences talk to the body's memory machinery. We used Python and a Python package called Brian2 to develop a brain simulation that maps neurons according to the Flywire whole brain connectome. Our simulation accurately maps neurons based on synaptic data from Flywire, and it can compare samples of gustatory receptor neurons, or GRNs. The 4 taste modalities that we analyzed were combined sugar water, bitter, sour, and IR94E, which encodes umami taste. We used the Zikevic mathematical model to model membrane potential dynamics. We found that from comparing the downstream neurons of both sweet sensing and bitter sensing GRNs, that there is equal overlap between sugar and bitter with sour downstream neurons, likely indicating that sour sensing GRNs have an equally appetitive and aversive valence, triggering a positive and negative taste response. In this simulation, of the sugar and bitter circuits that reach a memory formation associated neuropil, or location in the brain, at the 3rd order, all come from bitter GRNs, and only 2 circuits come from sugar GRNs at the 4th order. We approximate that this indicates an earlier aversive response for bitter taste than sweet taste, perhaps for the purpose of prioritizing avoidance of harmful foods over the encouragement of metabolic sources. Finally, we analyzed the number of synapses where either octopamine, dopamine, serotonin, or GABA were released, and found that in all 4 neurotransmitters, 3 of the 4 taste modalities were consistently correlated in their connectivity patterns. Similar synaptic counts for every neurotransmitter across all of the taste modalities suggests that taste memories, for the most part, use these neurotransmitters to the same degree and for the same function. There are certain outliers for this trend. What's interesting is that sour GRNs behave differently in octopamine levels, which is involved in reward responses, because they have both appetitive and aversive valence. And this may explain the very slow rise in octopamine levels in the graph in my poster. The sudden decline in octopamine for the 3 other tastes may not insinuate a lack of memory formation at that order, but a maneuver from a reward memory to a punishment memory. Our simulation does, of course, have some limitations. It does not represent feedback mechanisms or neuromodulatory downstream effects from certain neurotransmitters. But we hope that our simulation can be used to find notable motifs of neurons in future research, and that the accurate membrane potential dynamics from the Zikevic model can be analyzed. These results, while still being researched in fruit flies, can translate into understanding how we make dietary food choices, aiding us in research in dietary science and reward and punishment relationships with food. Lastly, I would like to thank Justin Ng, the Lu Lab, and the Open Bio Student Research Institute for providing so much support in my research. Thank you.
Awesome. Thanks for the amazing presentation, Annika. Now we're moving on to what I believe is the last presentation for this room, which will be Arjun. He's going to talk to us a little bit about transcriptomic dimensionality. Take it away.
Hi, good afternoon, everyone. I'm Arjun, and before I begin my presentation, I'd like to give my thanks to my mentor, Ankita Roy Chowdhury, and the Harvard SRI program for facilitating this project. Without them, none of this would have been possible. Cancer is often described by specifics, specific mutations in specific cancer types. However, general facts like the number of or percentage of mutations as a whole can actually tell us a lot about what tumors are doing. In this study, we examined one major type of DNA mutation called somatic copy number alterations, or scNAs, and how they affect tumor gene expression or behavior. In specific, if DNA is like a book, scNAs remove and add entire pages or chapters. scNA burden is the measure of how much of the book is altered, whether in a count or percentage form. Past research has linked higher scNA burden to increased tumor growth and proliferation and decreased immune response. However, this past research has often only measured a limited amount of genes, and furthermore, almost no one has investigated the structural effects of scNA or mutational burden on gene expression. In this project, I analyzed over 3,500 tumors from across 12 cancer types in order to gain a better understanding of how these DNA mutations shape tumor behavior and complexity. I began by calculating the scNA burden for each sample. Then I analyzed mRNA sequencing data, which represents the gene expression or what the tumor is doing, using a technique called principal component analysis, or PCA. What PCA does is it essentially boils down the over 16,000 gene counts per sample into interpretable major patterns, which I then linked to scNA or mutational burden through regression. Lastly, I investigated the links between scNA or mutational burden and the dimensionality of gene expression or complexity. I found that scNA burden is significantly associated with major axes of gene expression. As seen in figure 1b, in which the major axes of gene expression are shown, and also samples are colored by their scNA burden, you can see a small gradient, particularly in the y-axis from high to low scNA burden. We confirmed these relationships in figure 2, where we regressed these top patterns of gene expression against scNA or mutational burden and found that several of them were significantly correlated. Biologically, this confirmed prior research. We found a positive association with cell cycle and mitotic pathways and negative association with immune response. However, we also uncovered new relationships, such as with a positive one with microtubule transport and negative one with vascularization. These trends often persisted within within each cancer type themselves, as seen in figures 3 and 4. In figure 3, we show the significance of correlations between the top gene expression patterns and scNA or mutational burden within each cancer type. And in figure 4, we show the overall landscape of scNA or mutational burden within each cancer type as it related to the major gene expression patterns. As seen in figure 5, we further found that scNA burden is directly related to the dimensionality of gene expression. So what does that mean in more simple terms? Well, the more scNA mutations that a tumor has, the more complex of genes, or the more complex the structure of its gene expression, which means it's less interpretable into simple patterns. Lastly, we use partial least squares regression, a technique to find the patterns that most relate to scNA or mutational burden. We found that scNA burden was highly associated with increased intracellular transport and decreased immune response pathways. In this study, we uncovered several novel relationships between scNA burden or mutational burden and tumor gene expression or behavior. Why is this important? Well, first of all, it helps us get closer to a clearer understanding of how cancer is driven by these key DNA mutations, but also it unlocks potential further therapeutic opportunities. For example, if a patient has a high scNA burden tumor, we can prescribe them microtubule targeting agents, which are a class of drugs that attack intracellular transport, because we know that scNA burden is highly associated with increased intracellular transport. Some limitations of this study included not accounting for tumor purity and dosage effects, and also the linearity of PCA. However, further studies introducing more techniques and more transcriptomic analyses can further our research. In conclusion, our study linked large-scale DNA mutations to their effects on the tumor's gene expression or behavior, both improving our understanding of how mutations drive cancer and improving clinical therapeutics. Thank you.
Awesome. All right. Thanks for the amazing presentation, Arjun. And with that, we are officially wrapped up, I believe, with room 5 for SRI. I think most of the other rooms are done. I think there's still one or two left, but you guys all need to go back to the main room right now. Don't leave the Zoom. Go back to the main room. We have a very quick conclusion and just like one like logistics thing that Alex will take care of, and then we will let you go. You should be done in the next like 5 to 10 minutes, so hopefully it won't take very long. Looks like we're a little bit ahead of schedule, which is awesome, but please make sure to go back to the main room for very important stuff. Don't leave the symposium just yet, but you'll be out very soon. Congratulations again to you guys, and yeah, see you guys in the main room.